All right, on to chapter 2. Okay, so basically, chapter 2 is an intro to chemistry. Okay, so let's go through all this stuff. Okay, so everything's made out of atoms. Chemistry is the study of interaction between atoms. Okay, basically, an atom is the smallest unit of matter that can be subdivided into smaller substances and still be matter and still have the same properties. Atoms interact to form molecules. Okay, atoms consist of electrons, protons, and neutrons. So electrons are negatively charged, protons are positively charged, neutrons are not charged. And so electrons move around the nucleus. The nucleus in the center is made of protons and neutrons. Okay. The other thing that you should know is that electrons have no mass. Neutrons and protons do. Neutrons and protons have a mass of 1. Okay. Electrons have no mass. At least at this level of understanding, they have no mass. Okay. And protons and neutrons have equal mass. Okay. Technically not true, but close enough for this class. Okay. So atoms with the same number of protons are classified as the same element. Okay, they react similarly in chemical reactions. Okay, and therefore that number is really important, and that number is called the atomic number. Okay, the atomic number tells you properties of that element, and that atomic number is due to the number of protons. Okay, the atomic mass is something different. So neutrons have a weight of 1, okay, and protons have a weight of 1, electrons have a weight of 0. So what happens is you have different atoms with the same number of protons, so they have they're the same element, and they have the same atomic number, but they have different masses, and those are isotopes. Isotopes have different number of neutrons. Okay. Okay. So hydrogen, carbon, nitrogen, and oxygen are the 4 most abundant chemicals in living organisms. They consist of about 95–96% of living organisms are made out of those 4 and only those 4. Okay, elements. Okay, so make sure you understand those 4, the symbol for it, the atomic number for it. Okay, the approximate atomic mass for it. Okay, electrons are arranged in electron shells corresponding to different energy levels. Okay, so each shell has a different energy level. Electrons would like to be at the lowest energy level, and they form shells. Okay, so if I take a nucleus, I add 1 electron, it goes to the first shell. Second electron goes to the first shell. Third electron goes to the second shell. 4th, 5th, 6th, they go to the second shell. Okay, and then on third shell, 4th shell, etc., etc. Okay, and we'll get to what that actually means in a little bit. Okay, so make sure you can answer these questions. Okay, electrons want to fill their outermost shell. Okay, they fill their innermost shell and they go out, and ultimately they want to fill the outermost shell. Those electrons are going to be on the outermost shell are the ones that will be doing the interacting with other electrons. And therefore, they're going to be known as the valence electrons. Okay. Okay. Molecules hold together because valence electrons of combining atoms form attractive forces called chemical bonds between atomic nuclei. Okay. A compound is a molecule made of two more kinds of atoms. Is that right? Water. Okay. So the number of protons and electrons are equal in an atom. So if I lose an electron, I become positively charged. If I gain an electron, I become negatively charged. If I'm positively or negatively charged, I'm called an ion. Okay. All right. So one thing to go over with you guys is called the Octet Rule. Okay. I'm sure you guys have heard of it in high school chemistry. Okay. Atoms want to have 8 electrons in their outermost shell, with the exception of the first shell. The first shell has 2. The other ones want 8. Okay, 8 electrons in the outermost shell, and it's happy. Okay. So here you have sodium. Sodium has 1 electron in the outermost shell. So if it loses that electron, it loses that entire shell, and now it has 8 electrons on its outermost shell. Therefore, it is happy. Okay, so the last electron on a sodium very happily comes off, very easily comes off. Okay, so if you take that, and you take chlorine, chlorine has 7, and it wants to pick up an extra one. So it will grab an extra one. Okay, so now it has 8 electrons on the outermost shell, and it is happy. So that makes the sodium positively charged, and that makes the chlorine negatively charged. Opposites attract. Positive charges attract negative charges. They'll come together and make sodium chloride, salt. Okay? Okay. Atoms that are positively charged, or they lose electrons and become positively charged, are cations. If you gain electrons, become negatively charged, you're an anion. So in this picture, the sodium ion is a cation. Okay. The chloride ion is an anion. Okay. An ionic bond is a bond between a cation and an anion. Okay. So one atom loses electrons, which becomes the cation, and then it gains electrons, becomes the anion. These two are attracted to one another. Okay. So they are a ionic bond formation. Covalent bonds. Okay, so let me explain anions in a different way. The chloride really wants the electron. The sodium really doesn't want that electron, so it's able to just give it up and let the chloride punk it for the extra electron. Okay, that happens usually at the sides of the periodic table of elements. Okay. In the middle, where oxygen and nitrogen and carbon actually are, you don't actually get that. And so these things, these atoms kind of want more, kind of want less, but they're not willing to give any of them up. So they share. Okay? The other way to sharing is something like this, where these two are equal. So they have equal strength. So they pull with equal weight. Okay, so this is a covalent bond. They're sharing. From the point of view of this hydrogen, it has 2 electrons and happy. From the point of view of this hydrogen, it has 2 electrons and happy. They're sharing the pair. Okay. Here, carbon, okay, carbon has 4 valence electrons, and each of the valence electrons can share with 1 hydrogen. Okay, so those are bonds that form. So what you get is CH4. Okay, this is methane. Okay, all right. A hydrogen bond. Okay, so sorry, covalent bonds are much stronger and much more uncommon. It actually takes a lot of work to break a covalent bond. It's relatively easy to break an ionic bond. It's about 10 times weaker, if I recall. Okay, hydrogen bonds are even weaker than that. Okay, so okay, these are hydrogen bonds. Okay, so what happens is, okay, that's the hydrogen, or sorry, that's the oxygen. These two are the hydrogens. Okay, the oxygen is something called more electronegative. It wants electrons more, so it pulls the electrons toward it more. So now this side, okay, of the water molecule is partially negative. Okay, the delta means partial. So if this side is partially negative, this side is partially positive. Okay, so remember, negative and positive attract, right? So if I put another water molecule right here, the positive side, okay, this is positive, will attract the negative. Okay, and if I put another water molecule on this side, okay, the positive will attract the negative. And so each one of those bonds is called a hydrogen bond. A hydrogen bond is far weaker than the other 2 bonds, but they play a huge role in biology because they're incredibly common. And therefore, because they're so common, they play more roles. Okay? Okay. So let's go back to my picture. Okay? So those are hydrogen bonds. Okay. Molecular mass in moles. Okay, so the sum of atomic mass in a molecule is the molecular mass. So the molecular mass of water, H2O, each of the waters is 1, each of the oxygen is 16. So you add them up and you get 618. So the molecular mass of water is 18 daltons. Okay, or another way of expressing that, 1 mole of water, so 6.02 times 10 to the 22nd. You have to remember that number. 1 mole of a substance is its molecular mass in grams. So 1 mole of water weighs exactly 18 grams. Okay. All right. There are 3 basic types of chemical reactions. Okay. Chemical reactions always involve the making or breaking of a chemical bond. Breaking a chemical bond usually releases energy. Okay, forming a chemical bond takes energy. Okay, inorganic reactions absorb energy, so they require energy from other places. Okay, an exergonic reaction releases energy. They usually provide energy to other places. Okay, so you can kind of see how these two will fit together. An endogonic or exergonic reaction will feed an inorganic reaction. All right. The first type of reaction we'll go over is called a synthesis reaction. A synthesis reaction occurs when atoms, ions, or molecules combine together to form new, larger molecules. Okay, so these two come together to form AB. This is called anabolism. Okay, building of new structures. This requires energy. Okay, putting things together requires energy. Breaking things apart does not. So the opposite of a synthesis reaction is a decomposition reaction. Okay? Catabolism is a decomposition reaction inside a cell. When we break down sugar, it's a catabolic reaction. Okay? Exchanges are just part synthesis and part decomposition. You swap parts. Okay? The reversibility of chemical reactions can readily go in each direction. Each direction might need special conditions. The way you properly draw this, I don't know why it doesn't show up here. The way you properly draw this. Okay, so the way you read this, if you take AB, you add heat, you get product AB. You take product AB, you add water, you get reactants. Okay. Okay, so make sure you can answer that question. Organic compounds always contain two things, carbon and hydrogen. Okay, because carbon has 4 valence electrons, so it can form 4 bonds. Okay, so you can build huge structures with that. Okay, so organic chemistry is always the chemistry of carbon. Inorganic compounds usually lack carbon, and therefore they're usually small and rather simple. Okay. Okay, inorganic compounds, so acid, salt, base, pH. Okay, water, inorganic compound, polar molecule, which means there's an unequal distribution of charges, and we explained that already. Because it has an uneven distribution of charges, it is a very, very, very good solvent. Okay, salt dissolves in water very easily because it is polar, it's charged. Anything that is charged, salt is charged, sugar is charged, and therefore they undergo dissociation in water and they form solutes, and it dissolves a lot of things. Okay, like try adding salt to like oil; it doesn't dissolve very easily, or sugar to oil; it doesn't dissolve very easily. Okay, hydrogen bonds also absorb a lot of heat, okay, which makes it a temperature buffer. Um, it You can kind of oscillate the temperature, the hydrogen bonds a little bit, a lot actually, and that actually absorbs a lot of heat. So it takes a lot of energy to heat up water. Okay, so water maintains its temperature really easily. Okay, so this is the water molecule. This is the hydrogen bond. This is what happens when you actually add salt to water. The sodium chloride no longer stick together, and so they form solvent. These are called solvent shells around each molecule. Okay. All right. So acids, bases, and salts. Acids, okay, are things that add hydrogen ions to the solution. Like if you take HCl, okay, HCl is an ionic bond. If you drop it in the water, the H plus comes off and the Cl minus comes off. The Cl minus is perfectly happy with floating around. The H plus is perfectly happy to be floating around because they have nothing in the outermost shell, okay, which is nothing at all. But that adds hydrogen ion into the solution, and that is an acid. Now the hydrogen can attach to other things, so it is an acid. A base, by definition, adds OH ions. Okay. Um, this is slightly different definition in pure chemistry, but you don't need to know pure chemistry. This is microbiology. For microbiology, bases are things that add hydroxide ions. Okay. When you add an acid to a base, one adds hydrogen, one H plus, one adds OH minus. OH plus plus OH minus gets you water. Okay, so you add acid and base, and you get water, and then you get the components left over, which turn into salt. So the Cl from the HCl and the Na from the NaOH combine together to form salt. Usually the H plus and OH minus meet together and they generate a huge amount of energy. They release a huge amount of energy, which is why it's never a good idea to randomly add acids and bases together because things can actually go boom. Okay? Okay. I'm sure you heard pH before. Okay? pH by definition is the negative log of the hydrogen ions. Or hydrogen ion concentration. Okay. Um, Normal water has a pH of 7. Okay. Um, so normal water by definition has 1 to the negative 7 molecules of H plus. Okay. That is pH 7. If you put 10 to the minus 7 into this equation, out comes the number 7. So the pH of pure water is 7. Anything below 7 is an acid. Anything above 7 is a base. 7 is by definition neutral. Okay. Okay. So as you go up here, they become stronger and stronger acidic. Now they become basic. Okay. So there's a log of 10 there, which means if I move up a number, I'm 10 times as concentrated in hydrogen. So one drop of something that's pH 2 has 10 times the H plus of something that's pH 3, has 100 times the concentration of H plus of something that's pH 4, has 1,000 times the concentration of something that's pH 5. Okay. All right. So hopefully you answer those questions. All right. Functional groups. So functional groups, okay? Functional groups do certain things, like the hydroxide ion makes an alcohol, so methanol, ethanol, isopropanol, butanol, etc., etc. So this is a hydroxyl group, okay? Okay, there are certain groups that you have to know, okay? The alcohol group, the alcohol group you have to know. The methyl group you have to know. The amino group you definitely have to know. Okay. The last two that you have to know is the carboxyl group and the phosphate group. Okay. Make sure you know those groups because they are much more biologically relevant for microbiology than the other ones. Okay. I'm not saying these aren't, they really are, but you don't need to know that much micro. You don't need to know that much biochemistry. Okay. Okay. So two of the groups are the functional groups in this amino acid, right? The amino group and the carboxyl group. That's one of the reasons why you have to know what this is, because amino acids are really important. Okay. All right. So all right. Okay. Sorry about that. Let me fast forward to where we're going. Okay. So small organic molecules can combine into large ones. Okay. So now we're moving from chemistry to biochemistry. Macromolecules, the stuff that we usually deal with, are polymers that consist of large or small repeating units called monomers. Okay. Poly, many, mers, mono, one, mer. Okay, monomers are joined by dehydration synthesis or condensation reactions. Okay, um, condensation, you condense something, right? But I actually like the term dehydration synthesis better. Dehydration, removal of water, synthesis, put things together. Okay, this is dehydration synthesis. I am taking two components. I'm pulling a water out. See the water here, okay? And so I take the water out. That O directly attaches that R prime, and now I have R prime, and water. So dehydration synthesis. Okay, when I build things in cells, what I always do is dehydration synthesis. Now you can imagine what the next thing is. All right, so I'll come explain some more stuff later. So the building blocks of carbohydrates. So okay, so carbohydrates. Carbohydrates serve as cell structures or sugar energy sources. Okay, include sugars and starches. So carbohydrates always have the formula of CH2ON. So carbohydrate, carbon, so water and carbon. Okay, hydrated carbon. Okay, many carbohydrates are isomers. They're molecules of the same chemical formula but different structures. For example, sucrose and maltose and all these other things are literally just kind of the same chemical formula, but they're in different shapes. Okay, monosaccharides, mono one saccharide sugar. Okay, are simple sugars of 3 to 7 carbon atoms. Okay, disaccharide 2, right? So disaccharide is 2 sugars put together. I put things together through dehydration synthesis. If I want to break it apart, I go through hydrolysis. Hydro, water, lysis, break apart. I use water to break apart things. So here, okay, so this is dehydration synthesis. This opposite way where I'm going from right to left, that is hydrolysis. I'm taking water and I'm using it to break something apart. Hydrolysis. Okay. Polysaccharides, many sugars. Okay. Polysaccharides are formed through dehydration synthesis of mono and disaccharides. Okay. All right, lipids are the primary components of cell membranes. Okay, they are non-polar and they're soluble in water or insoluble in water. Okay, fats and triglycerides. So this is a fat. Okay, so fatty acids. Okay, see how there's very little oxygen here. Okay, there is no polar activity here, so the one side is not more polar than the other because there's no oxygen or very little oxygen, and so everything is equal. So there's no pull, so there's not polar. If it's not polar, then it's not dissolvable in water. So fats are not dissolvable in water. So these two are unsaturated. Sorry, these are saturated fats. Okay, these are saturated fats, which means every carbon is saturated with carbon or with hydrogen, which means there's no double bonds forming. Okay, this one is there is one double bond, so it is called a unsaturated fatty acid. The unsaturated fatty acid can either be trans or cis. This is cis. Okay, the two chains are on the same side of the carbon. Okay, like these two blobs. Oops, can't see that here. These two blobs are on the left side and not one on the left and one on the right. Okay, that is a unsaturated fatty acid. Unsaturated fatty acid can have a cis component. This is a cis, or it can have a trans one where they're on opposite sides. Trans ones look like saturated fatty acids. Okay, trans fat is very bad for you. Okay, okay, so cis fats look like this. You can't stack these things together very well because there's a big kink in the middle, so they don't stack. So they're not that close to each other. So they form liquids, okay? Or in terms of fats, they form oils. If they are all unsaturated and they're all stacked nicely in rows like this, and in rows like this, you can stack them on top of one another very neatly, like little boxes. And therefore, they're closer together and they form fat, like solid fat. Okay? And that's the difference. That's why some things are oils and some things are fats. Okay? Oils have more unsaturated fatty acids than fats do. Okay? Trans fat is particularly bad. Trans fat is a chemical way of turning a cis into a trans bond, and that makes it solider, makes it more closer together, and makes the molecules closer together and compact together, and they form fats instead of oils. Okay, if you have fat, it's easier to transport than oil. That's why they do it. Okay, that's why you get the partially hydrogenated vegetable oil. That's what they mean. They turn oil into fat for transport, and then all you really do with that is you melt it and it turns into oil again. So that's how you ship the trans fats. They have cut trans fats out of our diet because they realize it's not really processable and it collects in the human body, and that makes it bad. Okay, complex lipids. This is a phospholipid. It's part of the phospholipid bilayer. I trust you guys have anatomy by now, and we talked about this. Steroids this is a steroid. Okay. Okay, proteins. Okay, proteins are essential in cell structure, and this is what they do. So just read through that. Okay, proteins are made of amino acids. Amino acids have a carboxyl group, an amino group, and a side group. Okay, so there is always a carbon in the center. The name for that carbon is called the alpha carbon. Okay, on one side you have a amino group, on the other side you have a carboxyl group. Okay, and then on one of these you have a side group. The side group can be anything, and the amino acids, their side groups get mixed into chemical reactions a lot. So the side group of each amino acid is important. Okay, that said, you don't have to memorize what the side chains are. Okay, so you can see some of them are acidic, some of them are basic, and they can form different chemical reactions based on which amino group they have. Okay, you can skip this slide. DNL. Peptide bond is important. A peptide bond is formed between two amino acids by dehydration synthesis. So here, the carboxyl group and the amino group, they actually lose the water and you form a peptide bond. A peptide bond is a very strong bond. Okay, it's stronger than normal covalent bonds. It's in chemistry, it's like a bond and a half because it's literally partially this bond is down here as well. And so this peptide bond is a very strong bond, so it takes a lot of energy to break. Okay, proteins have structure to them. Okay, And the levels of protein structure, there are 4 levels of protein structure. The first level, the primary structure, is just the polypeptides put together. So here are the polypeptides. They're always written from N to C, okay? From the amino group to the carboxyl group, okay? That is the primary structure. The secondary structure is when you get hydrogen bonds forming, and they can form 2 separate structures. Helixes, okay, called alpha helixes, okay, and beta pleated sheets, or just pleated sheets, where there's a lot of hydrogen bonds. Remember I said hydrogen bonds were more common than anything? That's what I'm referring to. They form a whole bunch of hydrogen bonds. And so these are alpha helixes and these are beta sheets. They're the secondary structure. The tertiary structure is you combine the alpha helixes, beta sheets, and the other stuff to form a 3D shape of a polypeptide. Okay? The quaternary structure consists of 2 or more polypeptides put together. Okay? So make sure you understand the definitions of each of these 4 levels of protein structure. Okay? So primary, secondary, tertiary, quaternary. Okay? These things only exist well at certain temperatures, depending on the protein, and certain pHs, also depending on the protein. Like if I change the temperature, then this is going to, if I increase the temperature, it'll unfold. If I increase the acids, they will form different attractions to each other. So these primary, secondary, tertiary structures usually only form under certain conditions. Okay? And when we talk about it, we're usually talking about it being physiological conditions. Okay. So proteins can undergo denaturalization, okay? Which is what happens when you enter a hospital environment. Okay. So take an egg, you throw it onto a hot frying pan. What happens to the egg white? It goes from being clear to actually white. It goes from being gooey mixture to a solid substance. Okay, that is denaturation. Okay, when I denature something, I unfold the physical protein. I unravel the quaternary, tertiary, and secondary structures. I actually don't change the primary structure, okay, but I change the secondary, tertiary, and quaternary structures, okay, and they lose their shape, and therefore they lose their function. Okay. All right, we'll skip these guys. Okay, make sure you understand this. Okay, nucleic acids consist of nucleotides. All nucleotides consist of a 5-carbon sugar. So 5-carbon sugar is a pentose, pent 5-ose sugar. Anything that ends in OSE means a sugar. Okay, a phosphate group and a nitrogen-containing base, either purines or pyrimidines. Okay, nucleosides do not have the phosphate group. All right, deoxyribonucleic acid exists as a double helix. A's always bind with T's, C's always bind with G's. Okay? Okay. And look down the middle of the hydrogen chain. It is hydrogen bonds. A's attach to T's because they form 2 hydrogen bonds between them. C's attached to G's because they form 3 hydrogen bonds between them. And so these hydrogen bonds are what holds the 2 halves of your DNA together. Okay? RNA contains ribose instead of deoxyribose like DNA. That may not sound like a big difference, but it's huge. Okay? It is a very big difference. Ribonucleic acid is much more sensitive to acids than DNA is. DNA is much more stable. Okay? Okay, ATP is made of ribose, adenine, and 3 phosphate groups. Okay? It is literally made from RNA. Okay? So here is ATP. Okay? Okay. ATP is the energy currency of the cell. Okay? So every time I want to release energy, I undergo dehydrolysis, right? I hydro water, hydrolysis, break these two apart and release tremendous amount of energy. When I want to store energy, I take this and undergo dehydration synthesis. I add this phosphate there, I form water, and I get ATP. So higher energy molecule. Okay? All right, I hope this explains everything. I will see you guys for chapter 3.